Hey everyone, welcome to this week's broadcast of This Week in Science. Blair and I are here to talk about science. We've got some stories. Yeah, so as we get this show on the road, I mean, it's not on the road, it's on the internet right now. But as we get it going, I want to remind all of you that anytime that we mess up or have tech difficulties or things might need to be changed a little bit, not like AI changed or anything, but we got to get rid of a few oopsies. Our wonderful editor, Rachel, will take care of that. And the podcast is where you can find the edited version of This Week in Science. And otherwise, we're here for you now. It's showtime. Yeah, you ready? So,
oh my gosh.
Sorry. I need to change the way I put things in my life. I don't know what I'm doing anymore.
It was a good piece of Foley art, if that's what you were going for.
But yes, it's being taken out as we speak by our wonderful editor, Rachel. Okay. As we also get started, remember to hit the likes, hit the subscribes, get us into the algorithms. Let's get going on this show. Are you ready?
I'm ready. Let's do it.
I'm doing a science show. Yes, thank you, Paul Disney. Yes. I can be a totem pole. Okay. I'm a totem pole. Starting the show in 3, 2. This. Good. I do need to move that. Okay. I'll learn eventually.
Okay, give me a second. Hold on. Okay, I'm ready.
Great. 3, 2. This is TWIS. This Week in Science, episode number 1053, recorded on Wednesday, March 25th, 2026. Science Myths De-Fanged. Ooh. Is that like declawing? I don't know. Anyway, I'm Dr. Kiki, and today we will fill your head with cloning, hot plants, and platypodes. Or platypus. But first...
Disclaimer. This lovely show has been going for a long, long time. So long, in fact, that some facts are no longer facts. More than 1,000 episodes and more than 25 years means science has continued to progress. And so we have some fresh new science that is not so slightly contradictory to previous reporting to explore and learn about together on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science after a week off due to various things that got in the way of us doing this show last week. But we're back again, and we're glad that you're here with us as well. We have so much science news for you for this week. I have hot plants. I've got about, story about how humans think about how things sound in nature versus animals. So do animals and humans agree? More up at 11, Bob. I've also got some really interesting simulated flies and mouse vision. What do you have in the animal corner, Blair?
So that's interesting. I have flies and mice also, but different fly and mice stories. So that's really cool. I have cloned mice. I have courting flies. And then I also have the De-Fanged myth that I mentioned previously. We'll get to that. And then I also have the previously mentioned platypodes, which they were. So as an animal enthusiast, to say the least, I will say I've had many favorite animals over my lifetime. And the duck-billed platypus was one for a good couple years. So I got deep into platypus knowledge in elementary school, late elementary school for a couple
I'm really
years.
not surprised.
Yeah,
And
yeah.
I mean, the platypus, it is one of the most interesting animals on this planet.
And tonight, I'm going to tell you another way that they've discovered that they are crazy interesting. So I'm pretty
Okay.
excited about that.
I'm excited too. Oh, yay. So much fun stuff. I hope everyone is ready. We're going to have fun. We're just going to have a great night tonight. And I think I have to let you know, as we jump into the show, that if you subscribe to TWIS on any of those podcast platforms out there, it'll let you have TWIS whenever we release an episode, which is usually on a weekly basis because we live stream on Wednesday nights at 8 p.m. Pacific time-ish. I need to stop hitting this thing. Go
Put your hands in your pockets.
No, that's not, I am one of those gesticulating speakers. And yeah, anyway, I'll mute and I'll move it around.
There has to be a scientific study at some point where they restrained people's arms who are like who talk with their hands and then they suddenly couldn't verbally express themselves. That has to be a thing, right? I think if I were restrained, I would have a lot of trouble trying to talk.
Look, you just slowed way down.
There you go. Ask and answer.
But anyway, this sounds like a great graduate student research project. Let us know what your results are out there.
Mm-hmm.
We broadcast weekly on YouTube, Twitch, and Facebook. And so those broadcasts become the podcast. But if you are interested in just getting to our website and finding out information about the show, search for This Week in Science. Our website is twis.org. And now it's time for the science. Bum, bum, bum. All right. I'm going to start the show. Are you ready?
I'm so ready. Give it to me.
It's been weeks.
I thirst for science.
I know. I'm so excited. Hot plants. What do you think happens to plants when you make them hotter? So
what do you mean make them hotter? Like you talking about climate change? You're talking about I turned up the heat in my apartment? Like what do you mean?
Right. So it's interesting. We do turn on the heat in our apartments, right? And especially since the pandemic, people have all sorts of plants to plant with. And I don't think we see any changes to those plants that are living in our homes, even though we may have seasonal fluctuations that are, potentially outside of their normal temperature variation. But it's an artificial environment, right? We've got soil that we add fertilizer to, that we give plant food to. We make sure the plants that are in our homes are able to survive there as long as we're taking care of them. But what about the plants in the wild? And especially with respect to climate change, when we have been discussing an estimated 2 degrees centigrade increase in temperature on average globally. What does that mean to different locales? And so, it's like, okay, whatever. You have, equatorial plants that are hot and humid all the time, or dry and hot, but it's more sun, so more hot, right? Then you have plants that are maybe in deserts or that are more savanna-like, or maybe they are at the tops of mountains. The researchers for this study simulated a 2 degrees Celsius increase in temperature in a Rocky Mountain meadow to see what would happen. And so they were able to experimentally change the temperature of a meadow in the Rocky Mountains. And they're looking at these
set up a bunch of space heaters or what?
Right. Yeah. So like they really did use little heaters and
wow.
Adjust. Yeah. I mean, it was probably very energy intensive, but they changed the temperature of a meadow over time. And then they checked to see what happened to those plants over the time period of the study. And so when you think, Blair, of like woodland mountain meadows? Like
Mhm.
what do you imagine? What do you see?
Woodland mountain meadow. Well, the word meadow makes me think about like grass and flowers, of course.
Mhm.
Woodland sounds like some trees. Mountain makes me think there's some rocky outcroppings,
Mhm.
perhaps.
Yeah, and very often these meadows, they're a little bit grasslandish, right? But yeah, flowers are a big part of that. And so
Yeah.
you get like in Oregon on the mountainsides or even Colorado in the Rocky Mountains, tourists are there in the
Yes.
springtime to see the blooms of flowers
Mhm.
across the mountainsides, right? You can go on these hikes and see these amazing, sometimes super blooms, they call them. Like sometimes
Mhm.
now I'm like, everything's sensational. It has to be super this or
Yes.
super that.
Yes,
I'm gonna
working in a parks department, I can tell you that when there's a super bloom in a particular park and it goes viral, suddenly there are
everyone's there.
swarms of people taking their
People.
pics.
Yeah. And that's a whole other human behavior story.
Yeah.
But take that wild woodland mountaintop meadow and increase the temperature over time, over like where it lasts for a long period of time, over more than just one seasonal change, but over several years. What happens to the composition of that meadow? Does it stay the same? Is it able to adapt and exist with that 2 degrees C change, or does it have other issues? What they found is that there are issues that we did
Yeah.
not expect. Basically, a lot of these plants in the meadows have fungal like symbiotic relationships within the soil that allow them to get all the nutrients they need from what are potentially not incredibly nutritious rocky soils, right, at the tops of mountains. When they raised the temperature, they saw a transition from meadows to shrubs, sagebrush,
Mhm.
grasses, and wildflowers disappeared because the fungal populations, the bacterial populations in the soil changed as the temperatures increased because those microbes in the soil changed, the plants changed, and it
Interesting.
became sage plants.
How do they know it's
Yeah.
the fungus specifically,
They were
and
looking at
not something else? Okay.
Yeah, so this is exactly what they looked at. So they were, and I love this story because it came out of the Rocky Mountain Biological Laboratory where I actually spent a summer, and that's where
Whoa.
I was like became an ornithologist. This place is amazing. So if you're into like research and biology, and this is an amazing place. And anyway, that's an aside and a tangent. Gothic, Colorado, where this study took place. So they basically have this land that is uh, controlled, and they're able to have these long-term experiments that go on in this area because they own a lot of the land there.
Mhm.
Um, but what they found when they looked at the plants, at the soil, at everything, they found the surface of the soil was dried up by 20%. Shallow-rooted plants became stressed. Some wildflowers went extinct in the heated plots. And they also found because of that, the fungi and microbes in the soil also changed. So you have like this soil drying up. So you're not going to have fungal species that need the moisture anymore, right? And they found a decline in those symbiotic fungi that allow that nutrient capture.
Right.
They had an
Interesting.
increase in fungi that decompose organic matter. So basically, you had the heat, the soil drying, plants dying, and the fungal population was like, well, you don't need nutrients anymore. We'll digest you. And then that made way for sage plants and
Hmm.
shrubs. Yeah.
So there's some other things at play though, like if you heat up an entire section like this, that could also be a signal to the plants that it's a different time of year, right? and they could expect pollination when there are no pollinators. And so they can expend energy to attract pollinators at a time when they are not supposed to be doing that. And that can also cause extinction, particularly of flowers.
Yes.
So that's like another thing about, the timing of it. But I think that there's been a lot of studies on that, on like throwing the timeline out of sync for pollinators, for seed dispersal, for like releasing seeds and then there being another cold snap because it's too early, and then the seeds freeze and then they're toast, right? So like there's been lots of research on that, but I don't think there's been as much on what you're describing here, which is just it's hotter. It's not that the timeline has changed. It's not that the humidity has changed. Like a lot of these other factors have been studied. This is really just looking at hotter, and that's it.
Just locally hotter too.
Yes.
So yeah, and it isn't just a small time period either. Like they set this experiment up in 1991. And so this experiment has been running for 29 years, which,
wow.
Yeah. So it's a really long-term experiment in which they have been, putting space heaters, in a meadow, right?
Are they solar?
Probably. I don't know. I mean, I think that's a really great question. But one of the concerns though is whether or not a warming planet, especially on mountaintops, which are going to be some of the first ecosystems affected because of solar intensity and the changes to warming, not being close to, say, coastal winds or weather patterns. The issue is, how many of these mountain meadow ecosystems are just not going to be able to exist anymore as it gets hotter. Will they move further down the mountains, or are species just going to go extinct? Like there are many places around the world where certain plants only live in certain like mountain meadow ecosystems.
Yeah. I mean, this is the problem with mountain ecosystems in general. Is I used to talk about this on the Zoomobile all the time with chinchillas specifically. I would talk about it because they're from the Andes Mountains. They're up high. They're actually a desert species because it's a high desert. So it's like very low moisture where they live, but they're adapted to a particular temperature. It's quite cold. It's a cold desert. But so the idea is that as climate change encroaches on mountain species, plants and animals, the desire, if you were on level ground, if you were not on a mountain, would be to move towards the poles because you'd be chasing the temperatures you were used to. And it's basically the equatorial region is like growing, right? And so if you're in the northern hemisphere, the plant or animal would have a tendency to migrate north, and in the southern hemisphere they'd migrate south. But if you're on a mountain, you're going to go up in altitude because that's where it's going to be cooler. But as you go up in altitude, there's two problems. One, there's less oxygen.
Yeah, and that's another issue.
Yeah. And so that's why you see like pikas and chinchillas and other animals like guinea pigs are actually shrinking because of a lack of oxygen, which we've talked about a bunch. But the other issue is that there's less space and there's nowhere to go. And eventually you've reached the top. That's it.
Yep.
So it's
no more mountain.
Much more finite.
Yeah.
You don't have all the way to the Arctic Circle to move if there was land, but you have a much smaller geographic space to adapt, and it's hard for plants to move anyway, but it does happen to a certain extent with like seed dispersal and stuff.
Yeah.
But it is so much less room to move. The mountain species are really vulnerable. It's a big bummer.
It's like deserts, right? Because of these factors that you're talking about. And so what they're also considering is looking at the changes in ecosystems in other places as well. So on mountains, we're also seeing pine trees and other like more hardy plants starting to move up the mountains, like you were saying. And then places like the Arctic are also starting to experience this shrubification, where they do not. I love the word too. It's great. But the grasslands are making way for more shrubby plants. And so what does that mean for species richness, diversity, like so many things? Yeah.
Yeah. And once again, climate change all boils down to time scale, right? Like this sort of thing happening over eons, totally fine. Animals can adapt, plants can adapt. They'll figure it all out. Happening over the course of a few decades or a couple centuries, you're cooked. There's not enough time.
I don't want things to be cooked. I wish we could. I really want us to fix things. But, we're doing what we can with what we've got. Let's just vote differently, everyone. Okay, Blair, what did you want to talk about?
Yeah, so this was what my disclaimer was about. So every once in a while, a study comes out that is basically, hey, this thing that you've been telling people as like an animal expert for the past 20 to 30 years, it's wrong. So I have that for you today. This is the myth De-Fanged. I do believe I've talked about this several times on the show. And I actually recently met a rattlesnake expert through my workplace. Awesome. And they disabused me of this myth then. And then this study just came out, and I was like, what a perfect opportunity to talk about it. So as I have mentioned on this show before, for many, 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 many years, it has been understood, and the knowledge has been spread by biologists, by wildlife educators, by everybody. Baby rattlesnake bites are more dangerous than adult bites because the babies can't control how much venom they release.
And maybe it's you learn to control it and use it on prey species or whatever. Like you learn how to control it so you don't waste it.
Right. Yeah, absolutely.
Great logic.
Makes sense to me. Also, it means that if you get bit by a baby rattlesnake, the tendency is to insist that the medical care center you go to provide more antivenom. There is also a desire by people to dispatch, I'll say, baby rattlesnakes if they see them because they think they are more dangerous. And so there's a lot of weird repercussions to this belief in the end, when in reality, there appears to be absolutely no previous research to support this finding. And when they actually did research on this, it was found to be complete bunk. So this is a new study from Loma Linda University. It actually looks at the origin of this myth, the transmission of the myth, and the prevalence of it. And so this belief has, like I said, led to a lot of negative consequences. At the baseline, just fear of snakes in general has been higher because of this like mysticism of like, oh, if a baby gets you, oh,
yeah. Yep.
And so there's a lot of evidence now to show that baby rattlers, like adults, can control their venom expulsion. And in fact, adults possess and deliver more venom when biting, which also makes sense because they're going to eat bigger things. So they need to deliver more venom to the bigger things that they eat. So,
and maybe they look at something and go, you're really big.
Yes, exactly. And so also the studies looking at snake bite symptoms are more in larger snakes. So the symptoms are worse when they're larger snakes, which would also mean that if a human is bit by a rattlesnake, the adult does deliver more venom than the baby. Okay. Of course, here's the real disclaimer. If you are bit by a rattlesnake, or if it may be a rattlesnake and you're not sure, seek medical attention immediately. If you can, call 911. If you are out in the wilderness, see if you can get a medvac, because time is of the essence if you get bit by a rattlesnake. Now, that being said, before you go out in nature, if you can learn your snakes so that you don't get bit by a gopher snake and think that you were bit by a rattlesnake, that's helpful because a gopher snake is not going to hurt you at all. Wash it.
You're more likely to be defecated on. by a gopher snake.
That's certainly true. I've been bit by a gopher snake before. It did not even break the skin, also, I will say. But all that to say, seek immediate medical attention. You need anti-venom if you get bit by a rattlesnake. If you can get a picture of it, that's great. It'll help kind of identify what kind of snake it was. And also, if it was a rattlesnake, sometimes it depends where you were bit. They can deliver very specific antivenoms. So also, like, geolocation is important, letting people know where you got bit, all that good stuff. Anyway, that's my little, like, PSA about this.
I think it's
back
important.
back to the actual story, though. The rattlesnakes, also as a reminder, have an important ecological role, right? So we can't just go around eradicating rattlesnakes. We can't just go around chopping them all in half with a shovel, because they do have an important job to do. They remove a lot of pest species, a lot of species that can deliver disease to humans. And so they're still really important to their ecosystem. And especially if you're killing babies, then, like, they can't grow up and be adults and then have more baby snakes. So it's actually a huge impact on their population overall. So this is, like, especially important. But what they found through this research is that the myth started circulating in 1967. And this is from a California news media outlet. And this one media outlet seemed to be responsible for the majority of this myth getting out into the world. And this myth really circulated heavily in the 70s through the 90s. And then it was promoted by news media, like large news media, throughout North America from 2000 to 2014. These are prime Blair as a zoo educator years. So this is where I got this.
It happened. Like, why did people go on, like, a rattlesnake vendetta? Like,
I don't know.
It's amazing looking at the number of stories. It's not, I mean, maybe it was the internet and 24-hour news cycle and, the increase in just having to have stories everywhere.
Yeah. Well, also the 90s. Yeah, the 90s and early 2000s were also, I can attest, the era of, like, the TV wildlife guy, right? Like, there was Steve Irwin, there was Jeff Corwin, there were the Kratt brothers, there were all these different shows, but there were a lot of them, and they had to fill these shows with all sorts of fun facts while you were looking at an animal. And I also am wondering if some of this was just, like, somebody grabbed onto this and it continued. I mean, I bet if I dug out my handbook from my teen volunteer days at the zoo, I bet this was
got it in there. Yeah.
Yeah. And, I don't know what their citation was, but here we are. Right. But so they think that some of the original, like, sources, big air quotes, of this information came from news story quotes from healthcare professionals and emergency responders, like firefighters, police officers, and other rescue personnel. So not zoologists, nobody collecting data. And then quotes
from a
from
lot of anecdotes and people going,
Exactly.
This person was bitten by a baby and it was a really bad bite. This was an adult and it was okay. And so you're looking at the anecdote of who survived, who didn't, what happened to the wound, how much anti-venom was used. Like,
right.
Yeah. Okay. But not in a scientific way.
No. And in fact, when they
everything
had
went off.
quotes from more subject experts, what they would call them, including university professors, those were generally more accurate. So interesting stuff. The myth is so pervasive that 53% of the students surveyed in Southern California and 73% of emergency responders and health professionals thought it was true at the time of data collection of this study, which, again,
today.
I was telling people this all the way up until last year when I met a rattlesnake, like a real rattlesnake biologist, who came and did a safe rattlesnake handling class at my work, because we have rattlesnakes at quite a few of our parks. And I, like, geeked out with her for like 45 minutes afterwards.
Like, you got that as part of a training, but who gets that on a regular basis? Hi, snake biologist. Like, when do you get that opportunity?
Yeah.
So, Yeah.
Absolutely. Yeah. And she was like, yeah, that's a huge myth. I was like, oh my God. Let me buy your book, which I did. So anyway. And so part of this research was looking at how myths move around and grow, but also a big part of this was just trying to get the word out, because these snake researchers really don't want this misinformation to continue. There's a lot to lose by letting this misinformation continue on. And so I for a second I had some deep shame when I read this. It's like, how many people did I tell the wrong thing to? But
you were
we've
using
also
the info you had. That was
yes,
the most
absolutely
for you.
And we've also had things on this show where conventional knowledge has been overturned because of new scientific methods, because of new principles of understanding, because of new technology. And so, like, I just see this as another version of that, is that at some point somebody made a leap in logic based on observation, and it became tried and true knowledge. It became fundamental knowledge about an animal, which the more I learn about animals and the more I learn about things in this show, especially, the more I learn that a lot of the things that I told people on my tours through the zoo were BS. And so, you know, I can be sad about it.
And that's the way it works. You can't be sad about it. Like I said, that was the current like information, right?
Yeah. It's one of my favorite ones actually was that, not to go too far on a tangent, but I was a bear keeper for a long time. I also love to talk about the polar bears on my tours when I worked in education. And I would talk about how polar bears, when they go up to a seal hole, an air hole, to like ambush a seal that's coming up for air, they'll cover their nose with their paw because it's the only black part of their body.
Yes.
And I went to Churchill and they were like, what are you talking about? That's ridiculous. So like at some point, somebody said this thing and everyone was like, that sounds awesome. I bet they do that. And it just kind of turns into this game of telephone.
And that is exactly what happens. It like so much knowledge is a telephone game, right? Is just people picking up interesting things that make sense, right? That, oh, that's interesting. Or I really like that. You know,
but this is why you have to be a skeptic also. You have to hear things and go, wow, that's really cool. How do they know that?
I think that's the one, the skeptic, the question, how do they know that? Do we really know that?
Yeah.
I think that's the first one. The second is to be willing to bring new information into the way that you process, right? So that you learn a new thing and you go, oh, okay. And you don't fight and say, I have to keep saying that. And you don't get mad, right? You accept the information and go, wow, okay, I'm updating. And now I'll do better. Right?
I think, you know, not to get all political on us, but I think that's one of the fundamental differences between like the hardcore Republicans and Democrats, is that Democrats are generally, I'm talking in generalizations, there's a lot more opportunities to adjust ways of thinking on the Democratic side of the bench than there is on the Republican side of the bench. And so some people see that as weakness or wishy-washiness or like flip-flopping, but really it's incorporating new knowledge and making new decisions. It's like, and some people really have trouble with that. And I think a lot of the kind of fear of change on the Republican side feeds a lot of the kind of the digging in of the heels into the old way of doing things that causes a lot of the conflicts in American society.
And there have been studies, and I'm going to take it to a science perspective.
Yes, please.
There have been studies that show that there are genetic profiles and also brain profiles that fit into these different groups, and that additionally there are psychological identities that fit, right? So that the psychology of someone who's more conservative in the way that they approach life, not necessarily politically, but just the way they approach life, they want more certainty. They want to have you know, a hierarchical structure that allows them to know how things work. And it's like, you know, father figure kind of thing, right? Whereas being more quote unquote liberal in your life, you're a little bit more okay with uncertainty without needing it all to be just so. And because you're okay with more uncertainty, you can actually look at different perspectives and not be as challenged by them. Your identity is not challenged by that as much. Yeah.
Yeah.
Aside
totally.
from the other viewpoints that are like, there's only one way, right? And then you're like,
Yeah.
but that is not right. There is all these other ways. Look at all the ways we can look at things.
Yeah.
Which is, so it's these very different identity perspectives and profiles psychologically for existing in the world with other people socially and also with information. So yeah, scientifically this bears out. And unfortunately, and I don't like it. I hate boxes, right? And labeling people, but I don't know. We all choose. You're like, I'm this person. I'm that person. I don't know. I very often prefer to not be, I'm like, I'm not a skeptic. I'm a scientist.
Yeah.
Because I actually, because of certain people within the skeptic community, I have actually come to be like, I don't want to be associated with the skeptic community. I'm a scientist, so of course I'm skeptical of things, but that's just being trained to think scientifically.
Right. No, I think that's true. There's like specific types of skepticism that are just bearing on the ridiculous.
Yeah.
But there are other parts, there's the whole the fallacy of how do scientists know that, where there's never enough proof. Basically, you can just be like, but sometimes just said that there's gonna be another study next week, right?
Yeah.
So there's that piece of skepticism that is kind of unhealthy because you're never satisfied. There's never enough information. But there is the question of well, who's delivering this message? What do they have to gain by delivering this message? Was this study about climate change funded by the Koch brothers?
Exactly. Who funded it, right?
Yes.
Who did the study? Was it Heritage Foundation or was it, UC San Diego researchers? What is, where are you looking at things happening? Oh, wait, no, it apparently is not going to be our wonderful climate research station. That was the government. The one that they're breaking apart? It's terrible. It's the worst thing ever. And I'm so sad. National Research Institute on NRIC or something like that. I don't remember, but it's one of our mountain research stations. It has been one of the primary governmentally funded research stations to investigate climate change. And it's getting torn apart right now. Doesn't make me happy. But
yuck.
Yeah, but we can write our representative.
We'll rebuild. It's just
rebuild. It's gonna take a long time.
The lost time is very annoying, but we'll rebuild.
One step forward, two steps back. One step forward, two steps back. It is the way the pendulum swings. It's never even. Your study with the venom, though, that I think is so wonderful. 2015 to 2025. So we've got these years, 2000 to 2014, where it's all babies are more dangerous, venom dumping, blah, blah, blah. And then was 2015 the sea change of snake biologists getting when did you get your trainings?
2 years ago. So no. Missed me.
So interesting. But
I don't know.
I think it's interesting. It is a decade kind of clump that they've picked, or 15-year decade, whatever. But and it very well could have been more of a gradual shift, right? But I don't know. I think it's very interesting that there was a shift. And of course, because this is rattlesnakes, California is very highly, very prominent in the messaging, right?
Well, it's just, out of all the places you could run into a rattlesnake, we're the most populous.
Yeah.
There's more rattlesnakes in Nevada for sure, but there's less people.
And maybe Nevada is like, it doesn't have, because it's higher altitude, so much of it, like it doesn't have the same terrain. So the grasslands, the hills, the foothills in California, you go up there and it's in the spring, summer, like it goes from green grasslands that are like 2 feet high to just waving rolls of golden, right? But 2 feet high. And if you're running around your dad's construction site in the foothills, you hear this rustling that's just the grasses, and you can't hear the rattlesnakes. And so you have to like forge a trail through the grass and try not to get bitten by a rattlesnake.
You sound like you're speaking from personal experience.
I am speaking from personal experience, maybe just a little.
That's so funny.
Yeah. Anyway, I think I love going back and finding evidence that, and we can bring new information and new evidence to the fore. I am being visited by a 15-year-old
Hi.
Kai.
Hello.
Are you going to pull your head in here? You can't hear. Here's Kai.
Oh my gosh.
Yeah.
Hi.
Look at,
Wow.
Yeah.
he's
There we go.
tall and
Yeah.
here, microphone.
Yeah.
Are you going to say hi to everyone?
Hello.
Yeah, you cut your hair since the last time I saw you too.
Yeah.
Put your
It's
head in there. Yeah.
where?
Like you can see in the screen.
Yeah, I know. Like this. Yeah.
We're doing the show right now. Say
Nice.
hi to everyone out
Hello.
there. Hello, friends in the chat. This is the
Yeah.
child,
Yeah.
baby Nano
My
Kai.
Yeah.
goodness.
Actually, how long has it been since I've been like in the camera? It's been a while.
Yeah, 15
Yeah.
years.
Not 50 years.
How long since the last time? It's
Yeah.
probably been, oh gosh,
It's been
I
a
bet it's
while.
been 2 or 3 years.
Yeah, it might have been.
Because
Yeah.
you've stayed over there a lot and
Yeah, I
not
have. I
actually
usually
come in
just
the
like shut up
camera.
really quickly.
Yeah, people
Nice.
saying ahoy Kai, happy
Yeah.
Wednesday, hi Kai. Yeah.
Hello.
Hello.
Yeah.
Hey kiddo.
Yeah.
I love you very much.
Yeah.
I'll see you later.
Oh.
What did you take my knife?
Yeah, I'll take your knife.
You took my baby knife?
Take your tiny
You took my tiny knife. Now I'm sad.
He didn't want to say goodnight to you. He just wanted your knife.
He came in, he's looking around, and I'm like, what? And then he took my knife.
That was really shocking. I need a minute to recover.
Okay, so if this ends up in the podcast, I want everyone who has only been watching the show or listening to the show for a very short time to understand that Kai has been part of the TWIS experience and like all the stuff I've done online since before he was born. On the TWIT network, we did a monthly reveal of how big my stomach got, how much he was growing there. And then he was on camera all the time as a baby. And, when he was little, he'd pop in and he used to think he would want to do a show, but that was actually the most comfortable I have ever seen him. So maybe he's getting old enough to do a show. He used to get nervous, but
Oh my gosh. No,
Yeah.
I still
that's my
I still remember the
15-year-old
first
now.
time I ever came over to do a show and he was not yet walking. He was under a year old.
Yeah, and now he's taller than me. He is talking about wanting to get a job so that he can buy a Mazda Miata, and like it's amazing.
Oh my god. I need some time to recover, but we can continue.
We can keep going.
That's
Seriously, I live
that-
with him,
that-
and I need time to recover.
that
He keeps growing.
my
I'm like,
reality has been rocked by that. Like the last time I saw him was when I came to visit you. His voice
Yeah.
was in a different octave.
Yep, his voice has changed. He's, getting
He
muscles.
was a foot shorter,
He was
I think.
shorter. Yep.
Oh my god.
Yeah. No, the last couple of years have been whoosh. It happens. It's like a switch flips. And then they are like teenagers, like little adults. And oh my God. Yeah. Like he's ready to go. He's like, okay, ready to fly the coop, whatever. I'm like, wow. Yeah, everyone.
God.
So yeah, Thanks for enjoying that little sidebar with my son. Isn't that fun? It was so fun. He doesn't come down here all the time, so it's been a while. That was
Oh
fun.
my goodness. Yeah, we'll have to get him on the show.
Yeah, Derek, how is it that he's aged so much, but we're all the same age? I mean, come on. How is it? Yes. Yes, and we all have a fundamental misunderstanding of how time works, according to Fada.
Yeah.
Boy.
Wow. Okay.
Yeah. So anyway,
So yeah, what was
my
your
baby,
next
my baby
thing?
rattlesnake doesn't. He's not a baby anymore.
No. Yeah, tell me about sound.
I'm going to tell you about sound now. Yes. So when you go out in the world and you hear different noises, right? We listen to music. People like death metal or, bands like Guar. They also enjoy Shostakovich or Bach and, orchestral music, or they enjoy, rock bands or folk music, right? We have music. We also, and being podcast hosts for years, we've gotten I would really like your podcast, except I don't like the sound of your voice.
Yeah.
So there have been so many
Always.
of those comments, right? So audio is essential. Even though we as humans are eyes forward, so much of our brain is dedicated to visual processing. If your audio is bad, people will be less interested in listening to you. Audio is so important. We know what sounds good to us, right? We know what we like the sounds of. Some people like harsher sounds. Some people don't. Some people like softer sounds. You know what you like.
Mhm.
But so do animals. They know what is a mating call versus a danger call. They listen to other animals in their environments. And so the question that these researchers at McGill University and University of Texas, Austin worked on were questions about, do humans find the same things attractive that other animals do? Colors, sounds, etc. And so as experts in communication, they basically looked at a bunch of animal sounds that had been tested on animals previously and then got people to listen to sounds. And so they had an online game that over 4,000 people joined in and listened to different calls and said which one they liked more. And in judging and choosing their preferences, what they found is that people like the same things that animals do. The things that animals find attractive, people find attractive. We've evolved so, we've evolved so much above them. No, I'm kidding. That is not the statement. Don't take that. But we still like the song of a song sparrow versus the of a scrub jay, right? Or the things that we like, animals also like. People were faster in their study to tap sounds they thought were more attractive. And the fact that it was very fast, they think that there's a very subconscious aspect of processing and, that judgment aspect.
Were they judging the calls within the same species? And then this frog was the most successful, so this frog had the best mating call, and this frog's mating call was the one that was picked over other frogs? Or was it this type of bird versus that type of bird?
Right. So these were, with the animals, it was more, and it was frogs, insects, birds, mammals, and usually they had played sounds, and then the animals would choose one direction or another, right? And that was the animals choosing what they liked. And so it was a very similar test to the way they presented it to people.
Okay.
And so,
interesting.
Yeah, so I'm going to share this page from an article that was written in the conversation. And within this, let's see if I can make sure that the audio plays. Yeah, I want that audio to play. So this is a song that song sparrows, it is a song sparrow song, that song sparrows and humans prefer. And this that's not a song sparrow song. It's a different sparrow song. Song sparrows and humans did not like that one as much. And the researchers say that it's adornments, extra trills, fancy bits, chuck, chuck, blah, 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 you know, making it pretty, but in a melodic way.
Interesting.
These flourishes tend to make animals like them more, as well as humans liking them more. They did find, though, it's like a cross. So if a human was a musician, it didn't change anything. So people who were animal communication experts and musicians, what they were listening to in animal sounds, everybody kind of judged them the same. And additionally, they focused on sound in this study. And so the next is to look at color and other aspects of, what people like and what animals like.
Yeah, that's interesting. This is a tough one for me because I feel like there's so many weird variables. Like, which two calls did you pick? What is the relationship of those animals to the animals you're testing on, and the location where those animals are found, and the location that those humans are from, and like what kind of call was it? So like were they both the same? Were they both mating calls? Was one a warning call? Right? I feel like there's so many weird intricacies to this.
Yeah.
I don't know. That's interesting. But, from a sound design perspective, like when you're scoring a movie and you're putting sound effects in a movie, there are certain sounds that sound good and peaceful, and there are certain sounds that sound ominous. And so it also does make sense that there might be some sort of innate baked in response to some of these things. So I don't know. I feel both ways about it.
And some of their next steps also are to be like, okay, how do birds feel about frog calls? Which ones do birds like more? And do the ones that birds like, do those match with people? And I think part of it also is this really interesting idea of what, our auditory processing mechanisms are, where humans and birds, like, we have the same machinery in there, and we have a lot of the same components. But what about other species? So does it differ between species that have different ways of listening to sound? Right? Like, I don't know. I love the question. Why do we like what we like? And do we like the same things across the board?
Well, and then also, just really to throw a monkey wrench in the works, there are certain animals whose mating calls sound terrible to us, but obviously mean something to them. So, like, how would a macaw like these various sounds? And would they agree with humans? How about an elk? Like an elk mating call sounds terrible. Like, it sounds like he's dying. So I don't know. It's just I
have no idea. But yeah, so I think it's a very interesting question. But we have another question in this first part of the show that I think we really need to get at. How many times can we clone a monkey?
Great question. Well, this is about mice.
Oh, but still.
The picture, for some reason,
clone anything
is of a monkey. I don't know who put this picture on this
How many times can we clone Dolly the sheep? How many times can we clone a monkey? How many times can we clone a mouse? How can we
58.
clone? Why isn't it 42? Come on.
Weirdly, it's 58. Okay.
Okay.
So this is this is research out of Japan. They made 1,200 clones over 2 decades, starting with a single mouse. So why is the answer not 1,200? Well,
because different generations were different numbers of those clones.
So it was the 58th generation of mice had 0 viable offspring. And so this is the first time ever establishing that mammals cannot be cloned for an infinite number of times. So there goes the cloning route for immortality, which, like, is it even immortality? Because it's not your brain. You don't get your memories. So, like, it's anyway. Cloning is not immortality.
No,
it's no immortal jellyfish for me. Anyway, there were lots of hopes for this potential method of alternative reproduction, I guess I could call it, for the future, like saving endangered species via a seed vault type genetic depository of endangered or extinct animals, mass producing animals for meat that are all genetically the same. There's lots of, for research, medical research, everybody being genetically the same, that removes a whole mess of variables in your research, right? That could be really helpful. So there was previous understanding that based on how things were going, in theory, you should be able to create an infinite number of clones. But this was research that first started in 1997, which was a year after Dolly the sheep was the first ever mammal that was cloned. But in this particular study, the original mouse was born in 2005, which mice don't live 20 years. So this is pretty interesting that the same genetic material made it through 20 years. Pretty interesting. Once a mouse reached 3 months old, they were cloned again. And so they were able to get 3 to 4 new generations each year. And then over the next 20 years, they had over 30,000 cloning attempts, which only resulted in 1,200 viable mice. So already I'm going to say this success rate's not great. They removed the DNA, the nucleus of a cell from a donor, and then they implanted it into an unfertilized egg where they had removed the nucleus. And so that is how they were able to clone these animals. In the first few years, the success rate went up. At one point, it was 15% successful, which was considered good, I guess. And so they thought that they might be able to do this indefinitely. However, around the 25th generation, they started having problems. Harmful genetic mutations were building up over each generation, and each new set of mice had a lower survival rate. By the 57th generation, only 0.6% of the mice survived, and they were still healthy. They still looked just like all the other mice. They had no visible mutations, but the genetic code was wrecked. And so by the 58th generation, they all died shortly after birth, the ones that made it to birth. And so there were no visible abnormalities. There was no cause of death that they were able to identify, other than that their genome was it was bad. They sequenced the genomes of some of their clones. They found they had 3 times more mutations than mice born via sexual reproduction. They had larger placentas, and some of them were missing their entire X chromosome.
What? I mean, we talk about like the shrinking Y chromosome and that the Y chromosome is going to disappear. Nobody talks about the X. What?
Yeah, the X is missing. So the expectation was that all of the clones would be genetically identical to the original. That is not the case. Each time they did this, there was mutation, which, I'm sorry, of course. Like, you just popped a nucleus into an egg. Like, that thing still has to divide a bajillion times.
I think it's this is so fascinating from the perspective of cell division and, like, how that works. What are you laughing at?
I'm really confused as to why you would think there would be 0 mutations in this
So,
process.
yeah, so I think it's really interesting. Once upon a time, there was thought to be the Hayflick limit, where if you got a cell and you were cloning a cell line, that there was a limit to the number of times that the cell could divide and clone itself before it started to do bad things, right? And so with the study of the shoelaces, the aglets at the end of our chromosomes,
Yes, telomeres.
telomeres, thank you very
Yes.
much. Thank you for helping me there.
I forgot the word immortality earlier, so
Okay.
I understand. These things
So,
happen.
yeah, when we were talking about stem cells and cloning cells specifically, not whole organisms, the Hayflick limit was thought to be, it was like somewhere around 50 or 60 cell division generations. So I think it's fascinating
Mhm.
that it's 58 for the organism as well, because the Hayflick limit is, it's basically over time, the telomeres get shortened and shortened and shortened and shortened, even though the cell looks fine and the chromosome is still dividing and everything's great. And so it was all tied to telomeres. And then people are like, but wait, we can preserve telomeres and we could do this. And so there was this thought that we could
Mhm.
continue and have immortal cell lines. That because
Yeah.
the mechanisms that fix the telomeres were doing so continually, that it wouldn't have any other issues. Apparently, this does not work.
Well, they didn't try any interventions, right? This
Yeah,
was
okay.
really just clone, 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 clone. Let's see how long we can go. So,
But it's interesting also, the 25, like, it's almost halfway, right? To
yeah.
the 25 generations was where things started to go worse.
Yeah. Yeah.
Right,
So there's a clear turning point based on like, it was the carrying capacity of the ability to have mutations. And then it was like, all right, now this is a problem. We can't shoulder this anymore.
So, like, does that mean, though, if you understand that without intervention, that at 25 generations, that's when you go past the tipping point? But what if you clone, 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 and then do something to intervene at 25
Yes.
generations, and then clone, 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 right?
So this is a perfect segue into the next thing I was going to talk about. This is great.
Awesome!
So part of the study, they took later clones, some as late as the 57th generation, and they mated the clones with male mice, and they had healthy offspring with fewer mutations.
Got it.
So yes, you could in theory do this, which the first thing I thought of was all the like the whiptail lizards that do parthenogenesis. They'll every once in a while, they'll inject a little new
That's what
DNA.
sexual reproduction is for.
Yes.
Right? That sexual reproduction is, let's mix it up a little. Let's
Exactly.
add
Yeah.
some other stuff in there, and now we're not gonna go extinct. But
Yeah,
parthenogenesis,
so
parthenogenesis, parthenogenesis, oops, I need more
just a little
parthenogenesis,
salt
parthenogenesis.
into my I don't know. Anyway, but yeah, so they were able to kind of rectify some of this by just doing a little bit of old-fashioned reproduction.
Mhm.
So they're saying this discovery demonstrates sexual reproduction is indispensable for long-term survival of mammalian species. Yeah. It also supports a theory called Mueller's ratchet, which predicts in asexual lineages, deleterious mutations inevitably accumulate, ultimately producing mutational meltdown and extinction. So similar to what you were talking about. And so
no, mutations do not just happen because of sexual reproduction, right? Mutations are not just like crossover, change, blah, blah, blah. No, there are actual random switches. Single, the SNPs, right? Single nucleotide polymorphisms. Right.
Yeah, and if you're Yes. If
And it's like
you're
cosmic rays,
if you're
your
cloning over and over
chromosomes,
again. Yeah.
cosmic rays come in and go pew, 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 and then you're like, ah, I got a mutation. Most of the time we don't even notice.
That's right. Yeah.
Mhm.
Or, you get bit by a mosquito, gives you a little bit of epigenetic randomness. Next thing you know, you have a tail. No.
Hope not.
No, I took it too far. Well, so because I thought this is so funny. One of the things one of the researchers said was they joked that this finding meant that it would have been impossible to create so many clone troopers in the Star Wars prequel Attack of the Clones, which we had a whole conversation over dinner about in my household.
Of course you did.
Because we had this whole discussion about, well, they never really said that it was one sample of DNA that they took from Jango Fett and then they created all the clones from him. They could have taken his material multiple times. There could be many different lineages. But weren't,
there are different actors playing clone parts.
No, that's
there's diversity in there. Like that might be new canon. But
there's stormtroopers. So later they decided not to use clones anymore, and they started stealing children to turn them into stormtroopers. Really good.
Oh, right, because the stormtrooper clones could not shoot or aim.
Yeah. I don't remember exactly what they said.
Like dexterity and high-end.
Oh, I think Kamino was destroyed. Maybe that was it. Maybe the clone planet was destroyed, so they couldn't make anymore. I don't remember. I'll ask Brian. I'll get back to you later.
I bet they cloned and then fixed and cloned and fixed,
Yeah.
I mean, they had to make sure those midichlorians were happy.
Well, anyway, can't be cloned for forever, just 58 times. But
not even really 58 times. Like,
yeah,
57 was 0.06%. Like,
yeah,
come on.
30,000 times for 1,200 successes. That's not a good survival rate at all.
With diminishing returns after 25 generations. So come on.
So it's recombination through sex, very important.
I love it.
It's the reason we're all here, ultimately.
I think it's great. Studies like this are fantastic because it gets us to understand the box, right? The limits of certain technologies and what those limits are. And, hopefully will keep us from ever having a clone planet. This is This Week in Science. Thank you so much for joining us for another episode of This Week in Science. If you are loving the show, enjoying the conversation, please head over to twis.org and buy something using our Zazzle link. You click the Zazzle link, get some pretty cool stuff. If you're, it's springtime here, fall in the Southern Hemisphere. There are products that Blair has spent so much time and energy zhuzhing up in our Zazzle store that you can find things for the the Northern Hemisphere's summer that's upcoming, and even the Southern Hemisphere's winter. There are products all over the place. Pillows that'll keep you comfy as you hygge in the Australian winter, or maybe beach towels that say TWIS with our lovely, emblazoned burst that will keep you dry in the Northern Hemisphere's Floridian summer. I don't know. I just picked a state and a place, but you do you. Head to twis.org, click that Zazzle link, and find some stuff that's TWIS merchandise, helps TWIS out, gets you stuff you need or want for your seasonal activities or your just at-home activities, whatever it is. We've got stuff for you there. And at twis.org, our Patreon link. So if you are interested in helping us out in an ongoing fashion as a supporter, $1 a month, $5 a month, $10 a month, $1,000 a month. There are all sorts of options for you. $10 a month or more, and we will thank you by name at the end of the show. Yeah. There are so many ways. Please help TWIS continue and to pay the bills so that we can keep joining you and having nice conversations like this. We really can't do it without you. Thank you for your support. And now it is time for that wonderful time of the show we all love, known as Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Male fruit flies in love.
Oh, you must have a fruit basket.
Ah. So not really. They're not in love. Spoiler. But there is some interesting behavior happening with fruit flies that I want to talk about. So, Drosophila melanogaster, the fruit fly that is always studied in experiments around the world. I studied them myself.
You did?
Yeah, in actually in high school and college we did
Amazing.
Drosophila, whatever, melanogaster. We did research with them because they reproduce so quickly. They're so easy to keep, and they mutate really quickly, and you can do behavioral experiments on, like, there's so much they're so cool. Anyway,
Yeah.
this is not the point of this story. Let me back up. Okay, so
All right.
the melanogaster is like the classic fruit fly that is studied in labs all the time. And they are aggressive towards one another and will fight when competing for resources such as food or females. The males will, right? And in a particular lab, they were studying the melanogaster kind of fighting style. I don't know. That's a weird thing to say. Just the way that they fight.
They have aggression and they fight. Yeah. Yep. With their little legs and their mouth parts.
Yeah, and so they've been studying this aggression for decades. They've been looking at kind of what causes heightened aggression in the males. They've been looking at other things around it. And so most recently, they started looking at other species of fruit flies to see if the behaviors were similar. And generally, yes, except for one. Drosophila xanthomia, they have another tactic, which is courtship. So male to male courtship instead of fighting. Yes. So a senior postdoctoral scholar, research associate from the Anderson lab, Joe Wada, said, quote, all the conditions we've used to study aggression in other fruit fly species were the same. So we were expecting fighting, but these males behaved sexually towards one another. They courted one another all the livelong day. Man, Joe, all of your quotes are just, there's more coming. Okay. Even though they were set up in the lab by us to fight. So in this new study, as a result of that, they examined the underlying biological mechanisms for this same-sex behavior, or SSB, in xanthomia, and traced it to evolutionary changes in pheromones. So the pheromones are different in males and females of the species.
Right. And
There's
usually like stinky male, stinky female, and they like each other's scent and are attracted.
Right. And normally it's like, ooh, you smell like a dude. Get out of my way. I'm looking for a female. You're not a female and you're in my way. Right.
That's how they tell each other apart. One of the ways.
Yeah. And so in a lot of cases, if you see male-male courtship in an animal, it's often attributed to the idea that they can't distinguish males from females. Like this will happen like in some fish, there are males that won't get as big as other males. They look like females. The males will like court the other male, and then the male can sneak past the big guy and court a female, right? So it's like there's sneaker males, like that'll happen. But this does not appear to be what's happening here. It does seem like they can tell who the males and the females are. So they are, I'm going to say choosing, but they are intentionally, I'll say, courting another male. They can distinguish the males from females, And they're not trying to mate with the male. They're just doing courtship. They're just being sweet. And so this guy, Joe Wada, first saw their unique tactics when screening several different fruit fly species for aggressive behaviors. They were isolated for a week to prime them for social interactions. Then 2 random flies of the same species were put together in a chamber with a floor made of food substrate. In this case, it was apple juice. And the food, like exposure to a female fly, is a stimulating resource that causes fruit flies to fight. Quote, our idea was that once we found the biggest, most interesting evolutionary difference in fighting into different species, we would try to figure out what changed from melanogaster. And so once they started looking at this video footage, they found that they were doing the opposite of fighting. One of the 2 flies would raise a wing to a 90-degree angle and then vibrate to make a species-specific song, which is classic courtship behaviors performed by males towards females. So in previous studies, they showed that the pheromones do play strong roles in controlling fruit fly aggression and courtship. And so they did find that the pheromones on this particular species were substantially different from the ones that fought. And so it does seem like there's some sort of selective pressure on, they think it's a selective pressure on the females to prefer different pheromone type. Okay. So they don't have strong pheromones. And part of the reason for that is that there's an overlapping species of fruit fly where they live. See, this is getting like really deep. Sorry. But
so this is now like interspecies competition because they could do hybrids maybe?
So that's exactly it. They can hybridize, but to sterile offspring, like a mule, right? And so evolutionarily, you don't want to do that. So evolution is trying to push these 2 apart, repel them. And so the pheromone signals are becoming more and more different. And the way that they're becoming different, the male's pheromones are, for lack of a better term, less strong. And so as a result, the pheromones are not cueing in aggressive behavior. They're cueing calm, courtship behaviors. So even though the difference between males and females is large enough to this species that they can tell each other apart, the chemical signal that encourages other fruit flies to fight is not there because the pheromones are softer, different, more feminine. I don't really know what to say, but ultimately that is the leading hypothesis at this point.
Because if they are not competing with each other on a species level, they are ultimately competing against that overlapping species. They are all together ensuring the positive affectation or perception of their species.
Yes.
Right, of their species pheromone. By males and females. The question is, over time, and if you removed the other species, would aggression come later?
Right. And so that's the other piece is like, is this specifically fulfilling this function, or is there something else going on where because they're having this courtship behavior and not a fighting behavior, was that a happy accident that there's an evolutionary advantage to not spending energy and potential injury on fighting? Like, not fighting is the better choice evolutionarily. You're saving your energy. You're not getting a leg ripped off if you're a fly, right? So like there's a lot to gain by not actually engaging in physical combat. And so one of the things that they draw a correlation to is bonobos, which I actually think is a really good point, because you have your chimps that are pretty violent and you have your bonobos that are anything but. And so they have figured out how to work out social conflict, in some cases through sex, instead of through aggression. And so that is a really interesting parallel, I think, that they chose to pull here. It's pretty
and
neat.
the one thing you wonder about though is, okay, so the males are like, I like that smell, you smell good, let's be courting each other. But if they're courting each other, are they then not courting the females? And does that lead to an enhancement of female choice? Because when the females
Good
go, you both smell
question.
good, you're not fighting and you're both here, does the female then choose between the two, or does the female say you both are great? Like, so I think there's some interesting behavioral follows on
Yes.
this. Like,
Yes. So they're calling this, of course, preliminary results,
yeah.
and that the thing that they want to study next is if there is a benefit, of course, as we were talking about before,
Mhm.
but also we're calling it courtship.
Yeah.
And it
And it's
looks
just
like
not
courtship to
aggression.
us. Right. That doesn't guarantee that, quote, the male-directed form has the same intent as the female-directed form.
Yes.
For
Yes.
example, the intent could be to intimidate rather than mate. So they could actually just be, like how
Mhm.
deer will show off their antlers, their rack, and be like, hey, mine is bigger than yours. And they're like, okay, man, I'll back off. This is your zone, right?
Right.
So this could be like, look how good I dance for the ladies. And he's like, oh man, yeah, you have way more game than I do. I'm out. Right.
Well, it's
So
like
it could be that.
bro flies dancing for each other, practicing. Hey, ooh,
For
you look
sure.
good. I look good.
Yeah.
We smell good. They're gonna love us.
Absolutely.
I love
Absolutely.
it.
And so I have to say one more quote because it was my favorite one. Made me laugh out loud.
I love it.
Okay. Joe says, if you put two melanogaster males together, they'll fight. And if you put two male simulans, another fruit fly species found across the globe, together, they'll fight. But if I put my Santomia males in front of those other males, they'll all start courting my guys.
My guys.
So this pheromonal mechanism reaches across species boundaries.
I love it.
Joe Wada, do more research and please be interviewed
And be
always because I love it. I love your kind of tilt. I love the vernacular you're using. Like it's excellent. But yeah, I thought this was so funny, and I really did like the parallel to the bonobos. I thought it was a really cool example of how we do see this, because with the fruit flies, we always like to draw it up to kind of more charismatic species, I guess you could say. And so that is a really good example of a similar tactic working to a mammal's benefit, which is pretty cool. So anyway, make love, not war, everybody.
Yay. Yes, please. Can we?
All
That'd
right.
be great.
My last story for the night. I will keep it as brief as possible, but it's about the platypus. So it's complicated.
So I'm just gonna go away right now and you start talking.
No. I'm gonna try to keep it brief. So we've known about platypuses for a couple hundred years. The first platypus was brought over. Everyone thought it was a joke. They were like, it's a mammal bird because it had a bill on it, right?
It's a duck. No, it's not.
Yeah. It's a nothing. It's fake. It's a hoax bird mammal thing. Beaver. I don't know.
What is it? A jackalope?
Yeah, exactly. Yes. They lay eggs. They have venomous spurs. They sense electricity. They glow under UV light. They have more sex chromosomes than many other animals.
Except for the echidna.
Yeah, and one of my favorite things about it, I did a research project in college on lactase.
Yes.
In platypods, because they lactate. They don't have nipples. They don't have mammary glands like other mammals do, but they create what is milk. Is
it like a crop milk, but like in their pouch?
Well, it has lactose in it. It's not in their they don't have a pouch.
Okay.
but they have specialized hairs that are longer than all their other hairs, and the milk just kind of
What?
sweats out of their body and trickles down these specialized hairs. I wrote, I think, 20 pages about this protein
On this trickle.
found in platypus. Not about the function, it's about the protein. It was in my, this was my way of bringing Blair's Animal Corner to my microbiology class, because otherwise I found it very boring.
I love that.
And so I talked about
Yes.
platypus milk. Anyway, so that was one of my favorite weird fun facts about platypus. But interestingly enough, tonight's story about platypus is all about platypus hair. So related to those specialized milk hairs. You're welcome, everyone. I hope you're all eating dinner right now.
Hair.
It's about melanosomes. So melanosomes are specialized organelles in animal cells called melanocytes. Melanosomes produce, store, and transport melanin pigment. That is the determinant of skin, hair, and eye color. And it also serves other functions like UV protection, glowing in UV light, iridescence, other things. Melanosome structure is different across species and across groups of animals as well. They're usually different shapes and textures. They can be round, they can be rod-like, they can be flattened, they can be hollow, they can be solid. Hollow melanosomes are only found in birds until now. The hollow melanosomes are actually mostly attributed with the fact that the birds show up in bright colors and are iridescent. Like there are birds that are blue and green and red and yellow, and we don't have mammals that are those colors. And so the melanosomes are the reason that's understood for these extreme colors available in birds. So in mammals, melanosomes are round or elongated round. So like ovular, but they're always solid. And so all those other shapes and types are found in birds. The unusual melanosomes in birds usually have nanostructures that result in that iridescent structure. And so that completely changes kind of the spectrum of available colors for birds. What's interesting is that the platypus melanosomes are spherical and hollow. Hollow melanosomes have never been found in mammals before.
Wild.
Also, there's never been a hollow spherical melanosome discovered before, period.
Love it.
They looked in echidnas. They don't have these things. They don't have the spherical hollow. So like they can't find them anywhere. It's really only in the platypus. As far as we know, based on human sight, that's my asterisk here, right? These hollow melanosomes don't make platypuses iridescent, don't make them a bright color. They're brown. So it's weird for spherical melanosomes also to just be plain brown. And so the long and short of this is like, what? Why? Why do they have these? Why do they have hollow melanosomes? What function is that playing for the platypus? Why is it spherical and not any other shape? There's just like, there's one theory that it's ancestral because the birds and mammals do have a common ancestor a long time ago.
Yeah.
And so it would be possible that that's where it comes from. However, the fact that they're spherical and hollow and birds don't have those seems weird. Also, the fact that the echidna doesn't have it seems weird. The echidna could have lost it again, but it's probably more likely.
The sex chromosomes the echidna lost and brought back, like that there's losing and getting back again all the time.
Yes, absolutely. And so, kind of the Occam's razor of it is they probably developed this on their own for a reason. And so yeah, basically they don't know. The long and short of this is like, this is
They still
another
don't know.
super weird thing.
Even with
There's
this
like,
study, they don't know yet.
they want to look at genetic and developmental mechanisms on how they form. They want to do comparative studies with birds and other vertebrates to look at evolutionary pathways to these melanosomes. They think they'll be able to figure it out. The study authors say, excitingly, over 200 years after its description as something between birds and mammals, we find additional convergence between the platypus and birds. The discovery of a unique melanosome morphology opens up exciting new avenues of research into melanin genetics, melanosome development, and the evolution of melanosomes. So, yeah, this is very preliminary. I think they'll be able to figure it out. Like, this is definitely something where you can kind of, through trial and error, create artificial materials with different types of structures and try to see what different tweaks do. My money's on the fact that we are only seeing human sight.
Yeah. So you mentioned that the platypus also has ultraviolet spectra. Like
Yes.
yeah, birds do as well, not just their
Yes.
iridescence. And this is also something that like squids and other animals, like ultraviolet is, I think, a very fascinating thing. And if we're talking about the spherical structure with it, like a donut, as someone saying in the chat, with these melanosomes, what you have are structures that are they're physical structures that light hits, and then you have a certain refractive index in the structure, and then it hits the molecules within the structure, right? And so there's different amount of light emitted or absorbed. And if you're talking about a sphere, like how maybe the shape and the structure, maybe it comes out brown, right? Because they look brown
Right.
to us.
Yes. Yeah.
But because of the way that the donut works or something, maybe that gives the ultraviolet spectra. Like I'm totally spitballing here, but
Yeah.
I
No,
mean,
for sure. So there,
structure
yes.
and function, and if these are the things that give the color, right? So these are the shape and the molecules that are involved. They are absorbing and emitting light and allowing like certain things, refractory things to happen inside. There's interesting stuff here. I'm gonna guess.
Yeah, no, for sure. And that's, it could be UV. There's lots of mammals also that emit UV now that we know. Well, emit is the wrong word, but like
Mhm.
that show up a color under a black light that we
Yes.
wouldn't expect, right? That are pink. But there's that pathway, but there's also just like they could just be showing a color on a spectrum. We have absolutely no idea. There could be some sort of way that it plays underwater. You mentioned squid, so like, is
Mhm.
there something to do with when it's wet? Because
Which
not
the
a lot
platypus
of birds
spends
are
time in
not
water, right?
wet. Yeah.
So should we be looking at waterfowl
Right. Yeah.
or some of the species like penguins or
Mhm.
other, I mean, where else should we be
Yeah.
looking? I think that's
Yeah.
fascinating.
Yeah, because just because we can't see it doesn't mean that they're not some crazy color, or they don't have some crazy pattern. Like, remember humans are striped?
I am
Remember that
not.
whole thing?
Yeah.
Um, no, but I think that I love that there's just another thing that platypuses are just so weird. Like, what
Mhm.
happened to you guys? You just went off on a limb and just kept going. And because you're in Australia, there weren't a lot of large predators, and
There's
Australia was
all
just like,
sorts of predators, but
right
they just
on.
were in places and they have, their venom also. Don't they have a venomous spur or
Yes,
something?
they do.
Like the
Yes.
platypus is like,
Yeah.
got you. Haha,
Yeah.
you thought you'd get me. And then they go and they filter feed and stuff. And what the heck?
Yeah, they
And
can
then they
sense
and then they
electricity so they
yeah,
can go get their worms in the dirt. Yeah.
which is amazing. Yeah,
Yeah.
they're like sharks, but
Yeah.
not. And then they're in
Yeah.
the water. And then on the land.
Yep. Yep. And they lay their little eggs. Yeah, they're runaway. They're runaway evolution that works. And if anyone was curious, platypi is incorrect. You may say platypuses. If you really want to get Greek with it, which is the origin of it, it's platypodes.
Which
So
I
there you go.
love, because I went down an octopus rabbit hole related to that.
Yes,
many years ago.
octopode is also correct.
It's correct, but it is not the octopus is more
Yes.
correct.
Yes.
But this case, platypodes,
Yes.
platypodes,
Yeah.
yeah, they're great. I
There you go.
love these little animals. Oh my gosh. Thank you for the story.
Of course.
It's just new fun related to the platypus. Here we go. And you said you had, at the beginning of the show, we were like, oh, you have a fly story, fruit fly story? I have a fruit fly story. So can I follow on with my fruit
Yes.
fly story?
Please do.
Yes. Okay. So once upon a time, 2004, researchers published their model of an artificial simulated fly. So they used quote unquote artificial intelligence, a company called Genelia, and then also scientists from DeepMind. They created a virtual fruit fly, and they created a whole program for it, and it was called NeuroMech Fly, where instructions are given based on kind of what we understand of the neural net control model of the fruit fly brain, and how it controls the movements and flight of a fruit fly. And they started with NeuroMech Fly version 1, then their NeuroMech Fly version 2. And so 2004 is when they started. 2025, last year, they got into their version 2. And this virtual fruit fly, the research has been published in Nature Methods. It's been, all over the place, but it is effectively a morphologically correct digital twin. So the body is right. They think they've got the kinematics right. They think they've got all the control factors right. And if you were simulating a fruit fly brain and said, these are the parts of the brain that go up and down and over and blah, 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 blah. If a predator comes to smack the fruit fly, the brain of a fruit fly does these things and it should fly this way. So in their artificial simulated virtual fly, they've given it all these commands, and look, it moves and acts just like a fruit fly. Isn't that amazing? So this apparently, obviously, is going to tell us how and why the fruit fly brain works the way that it does. Of course, because when you create something based on assumptions with artificial intelligence and then use it in a simulated artificial environment digitally, that of course has real world applications and understanding. Sorry.
So I'm guessing not.
Anyway, so it's been published in these big places, and they, the model's brain processes visual and olfactory information. They've got like virtual eyes and antennae. It's basically they're like giving their digital twin fruit fly as real of a fruit fly experience as they can. And a new study was just published this week that I think is very interesting. It's not completely, it's a bioRxiv study. And so this means it is not yet peer reviewed. It has just been released. It's called The Digital Sphinx. Can a worm brain control a fly body?
Whoa.
And so in their short publication, they took the NeuroMech Fly simulated model and they said, instead of a fly brain, we're doing a worm brain. So they took C. elegans, all of the simulated all the information that would be involved in moving a worm, and they put that into this digital twin of a fruit fly instead of the fruit fly brain.
Mm-hmm.
And it worked fine. It was great. it did physically realistic walking. It connected really well. Like they had like a whole connectome, and they showed how in their model it all connected.
The walking's
They
interesting because worms don't
Yeah.
got
Right. Worms don't have legs, but in their model and what they showed in this digital twin that was, a digital hybrid amalgam, um, it worked just fine. All of the controls that are in our understanding of a C. elegans connectome and control matrix, it interfaced with the fly body model perfectly. Proprioceptors, actuators, so this is what it feels like, input, output. When it comes down to the level of just inputs and outputs and in this simulated virtual environment, everything worked the way that it does in the fruit fly model. And they could not tell the difference between a worm driving a fly and a fly driving a fly in their virtual, quote unquote, digital twin. Fruit fly getting a real world experience. Oh my gosh. And I
Well,
think
then
this is
send that fly into court with another fly, then you get into trouble, I think.
right. But I mean, I think the question that they bring up that I think is so apropos to the time that we're in and what's happening right now. Basically, the question of what value this simulated digital twin research is to us in taking it from the simulated environment back to real world biology. They question whether or not there's actually an application for that.
I was
Why
hoping
would
that you were
you?
going to tell me that they plugged a real worm into the fake fly.
When I first started reading the story, I was like, what did they do? Is this like real biology
like VR
experiment?
for a
Yeah,
worm.
did they take C. elegans brain cells and put them in a fruit fly? No, they did not. This is completely digitally simulated. It's not that exciting, but
So
what
they
they
trained an
say,
AI to act like a brain of a fly, and then they trained an AI to act like the brain of a worm. So there is an opportunity for some drift here, correct?
there is opportunity for drift, yes.
Okay.
But what they are saying is if you have that at some level in these simulated digital animals, if you just have a worm, right? A worm brain in a worm digital twin, it has sensory inputs and sensory outputs that lead to, proprioception. What does my world feel like? And then activation, actuators. How do you move, right? They put the exact same things into flies, except there are limbs versus
Mhm.
worm smushy. Um,
Worms.
and the bottom line, though, is that in the digital environment, it made no difference because inputs and outputs mapped regardless.
Right. They there was no sensory like feedback because of that, right? Like so because it was all
But it's the
digital.
same. So you poke a worm, you side by side, right? You have
Yeah.
a fly and a worm, right? If it's digital fly, digital worm, and in the simulated thing, you poke both of them, each of them are going to try to move away from the poke, right? So that is proprioception input, actuator output. Different muscles, right? It's different controls, but input and output is the same. And so then they took the inside controls of the worm and replaced the inside controls of the fly. But because input and output is essentially the same goal in the end,
Mhm.
poked,
Mhm.
leave, it all worked together. And so the researchers are saying there is a huge peril to using these connectome body models in the simulated virtual environment, because while you can show the behavior with fidelity, right? So poke, run away, there is not actual biological fidelity.
Hmm.
So it looks the same.
So did they
but
just
it's
tell on
not.
themselves? Did they just say like our research means nothing, basically?
But yeah, so this is an outside group who are like,
Oh,
we're
okay,
gonna we're gonna take this thing and we're gonna put a worm in a fly. And it did the same thing. We couldn't tell it apart.
I
So
see. Okay, yeah.
everybody's over interpreting the importance of this previous research. Like, let's
Yeah.
take a step back and talk about how we design these
Right.
synthetic models, right?
Mhm.
Why are we doing it in the first place? Do you just want like better virtual fruit flies for the Matrix? Like,
Right.
yeah.
Man,
So
weird.
I just love though when I find the papers that are not just following the bandwagon, but trying to poke the holes that are
Trying to break
necessary.
the wagon.
Because that's part of it, right? It's like you have to try to see, okay, you're doing this stuff. It seems to work. Where does it
Yeah.
break down? What have you overlooked? What are we as a field overlooking? And how can we use this to move forward better? You know, and if we want
Mhm.
to like actually have real fidelity between digital and biological worlds, so far we're doing it wrong.
Mhm.
So anyway,
Yeah.
we're doing it wrong, everybody.
Yikes.
Yeah, but anyway, that was my fruit fly study, and I think I got a kick out of that one. And Fada sent me a story this week that I thought was so great. Researchers published in eLife this last within the last week. Their work, we've talked about it many times before, of recording visual input from the brain of a cat or from the brain of a mouse or even from the brains of people, trying to have a digital reconstruction using neural signals from within the visual cortex to actually be like, okay, we can reconstruct this, which for animal models is really cool because we can't talk to animals and be like, tell me about your dreams. You know? And so if we can create a really robust, replicable, like digital model way of measuring animal brain activity while they sleep, maybe we could like watch their dreams. Maybe we could, if we can understand also what animals think, like what the brain looks at, like what is important in the reconstruction of the world in the brains of different species, predators, prey, running animals versus like static animals. Like there's so many different ways, like we talk about just between individual humans about the differences in the way that we could potentially be perceiving our visual environment just because of individual brain differences. But what about between species? Right? So like even a bird of prey who has like an amazing visual field,
Mhm.
like high resolution, super fast refresh rate, who can be like from like a mile up in the sky and come down and get a mouse in a field, right? Or the owl at night that is following that ultraviolet like trail of pee from behind a mouse or a vole in a field. What are they really seeing? And how is their like everyday experience, how does that work in their brain? And is it the same as the way we see?
Hmm.
Or is it different, right? How does it reconstruct? And so I never like I've always thought about like the reconstruction of what is the neural activity in the brain for vision. And I'm like, okay, that's cool, whatever. But like to think that not every brain is going to show like the movie reel that we expect as humans, right? Or like this consistent thing, right? Like we perceive this unbroken chain of visual input when in reality there's little breaks in the middle and it's like it's not perfect. It's a film reel, right? There's like
Well, and we learned some people think in pictures and some people think
And
in
some people-
words.
words. Hello. Yeah. It's like I have the concept, but if I say it, as soon as I say it, it becomes more real, but I still cannot see it.
Wow.
But I know
That's
what it
so
is.
interesting. Yeah,
I know
yeah.
what it is. Yeah, it's so weird. But animals don't have words like us, right?
Mhm.
So how do they conceptualize things? Do they have differences the same way we do between like more vocal, verbal members of their species versus others? And so anyway, I think it's a very interesting study, and they've finally gotten to a point where we're able to do higher resolution imaging of animal brains. And in this study, they did back propagation through neural encoding that they reconstructed. I'm going to sneeze. Okay, that's better. They reconstructed 10-second movies at 30 hertz, which is slightly faster than like the 24 hertz or frames per second of movies. And they used 2 photon calcium imaging data, which is very high resolution modern imaging technology. And so they were able to get this increase in not just like they're doing single cell recording basically of hundreds of neurons within the visual cortex of mice. And so they were able to like basically get these really cool kind of like low res pixelated images out of the mouse brains. And in what they did, like they've got some really cool reconstruction of these brains, right? Like I'm gonna try and open this image and then go to a different. Where did that story go? La 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 la. I put things places and then they went away. The researchers were able to. They started with just grayscale in what they started measuring. Like they showed gray images to the mice and then measured what the brain did and those cells did. And then they started mixing it up, adding more detail and all sorts of stuff until they were showing them little movies. And they have images that are dynamic, that are these 10-second movies they created, reconstructed from what was shown to the mice and then what was reconstructed from the neural activity of the visual cortices. And they're not the same. And it's not necessarily that, oh, this is a weird glitch in the system. They are looking at it and saying it's not the same because this is the way this mouse brain is dealing with this visual information. And so there are breaks in certain places. There are like the important, like more cells focused on certain things than others. Like the way it's reconstructed is very different from what they would potentially record from a person if they were doing the same thing. And so eventually the hope is that maybe we can actually record the dreams of mice and know what they are dreaming of and what is important and why.
Man, this feels like science fiction.
Yes, I love this stuff. This kind of stuff is like it because it's interesting, like this can be very useful and doesn't scare me. Whereas, oh, science used badly, for but this seems like something that could be such, I mean, and if you imagine also being able to understand in humans how human brains react or dream. Imagine being able to record the dreams of people who are minimally responsive or in comas, and there is brain activity in some of these people, and if you could just record it in the right way, could we actually communicate better? Could we know what they are experiencing while they're away from us? I don't know. I find the optimism for this one It's more on my upscale. It's good.
There's lots of cool applications. There's also like the
Mhm.
nightmarish one where people can spy on your dreams, but we'll leave that one. We'll leave
That's
that one in the dust.
future. Yes. But the idea also is, right now, this is like 2-photon calcium imaging is not something you can do in your bedroom. So,
Fair.
so
No eternal
there's a ways to go.
sunshine of the spotless mind. Yeah, no, absolutely
Yeah.
not. That's
Yeah.
funny.
Anyway, reconstructing the dreams
Wow.
of mouse brains, and they're getting better at it, and it's pretty cool.
Cool. That's awesome.
And to do that with birds or frogs or fish or whatever, how many different species could we potentially begin to get a glimpse at their experience
Mhm.
of the world? Like, I think that in itself is very exciting because we anthropomorphize everything. And it keeps
Yeah,
us
could we put a fruit fly on the stand at a murder trial because it saw something?
right. Oh my gosh, who needs nanny cams? It has to be within what, the 72-hour lifespan? No.
Yes.
On Friday, this nascent fly saw you murder. It's now Saturday. We only have 12 hours to continue talking to this fly before they go into.
Yep.
Oh my goodness. Have we done it?
We've done it.
We make it to the end. Thank you, Blair.
Yeah. What a fun show.
It was fun. I loved all the animals and the crossover between things, and how fun. Thank you.
Yeah.
Thank you, everyone, also who has joined us tonight. Thank you, those of you who join us later in the future, for romping through the lands of fruit flies and mice and science. It was a lot of fun. I want to give some big shout outs to people who help with the show on a weekly basis. Fada, thank you so much for your help with show notes and social media and everything that you do. And that story was fantastic. Thank you so much. Gord, Arnlor, others who help in the chat rooms. Thank you for keeping the chat room a wonderful, respectful place. And all of you who are there in the chat room right now, thank you for chatting. Thank you for being a part of this conversation and this show. It's always good to see your comments there as we're going through. Identity 4, thank you for recording the show. And Rachel, thank you for editing. And of course, our Patreon sponsors, thank you so much too. Aaron Anathema, Adam Mishkan, Alan Viola, Alan Coffin, Andrew Swanson, Artem, Arthur Kepler, Bob Calder, Bob Coles, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landing, Craig Potts, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Don Mundus, Eode, Eden Mundell, Eric Knapp, Flying Out, Fred S104, G. Burton Lattimore, George Corris, Greg Briggs, Jack, Jason Olds, John Atwood, John McKee, John Ratmaswamy, Johnny Gridley, Ken Hayes, Kent Northout, Kevin Parachon, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessenfo, Mark Jenkins, Noodles, Patrick Pecoraro, Paul, Pal Disney, Paul Ronovich, Philip Shane, Pierre Velazar, Richard Badge, Robert Norland, Robert W. Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean Clarence, Lamb, Shubru, Stephen Alberon, Steve Leisman, aka Zima, Sue Doster, Teresa Smith, Tony Steele, and Vegard Schefstad. Thank you all for your support on Patreon. And if you are interested in helping the show out, supporting us on Patreon, head over to twis.org and click on the Patreon link. On next week's show,
we will be back on Wednesday at 8 p.m. Pacific Time, broadcasting live from our Twitch, YouTube, and Facebook channels.
Oh, yeah. If you want to listen to us as a podcast, just search for This Week in Science wherever podcasts are found. If you've enjoyed the show, please get your friends to subscribe too. The more, the merrier.
For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twis.org. You can also sign up for a newsletter.
Someday. We love your feedback. If there's a topic that you want us to cover or address, a suggestion for an interview, let us know. You can let us know on one of our social media accounts or send us an email. Make sure that you put TWIS in the subject line so that your email doesn't get spam filtered into where, Blair?
A clone of 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 an email that you can't recognize anymore. Too many mutations.
I love it.
We look forward to discussing science with you again next week. And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science.
This Week in Science. I even wore my This Week in Science t-shirt today. Boom, boom, boom. How you? You're muted. They're not.
Do you want to re-record the very beginning just because you said 10:53?
Right, because I didn't update because of last week, and it's still, I just used what today's run sheet was.
Yes, of course.
That's what happened, everyone. I mean, I was going to just comment on it during the show and just be like, that's it. We're now at 10:53. Who cares?
It's up to you. Either way.
Yeah, no, let me, I just need to record the very beginning.
Yes,
right?
just this is TWIS, yeah.
Yep. I don't think I need after, just after that. Okay, Rachel, if you're still listening, this is what you need to put at the top of the show. So 3, 2, this is TWIS. This Week in Science, episode number 1052, recorded on Wednesday. No, is it 1052?
Yeah,
It's
I fixed it.
1052.
Yeah, I
You
fixed
did.
it.
Okay, good. All right. I got scared for a second. I'm going to go up there. 1052 in bold over there. I got to have my redundancies. 3, 2. This is TWIS. No. This is TWIS. No, I don't even know how to do it anymore.
This is TWIS.
Thank you. I'm too tired. Oh my God. I have to get up so early tomorrow
I'll just
morning.
do it. Ready? I'll do it.
I got to do it for the Science Talk conference tomorrow. I'm so tired and I have like slides to make for people and stuff. I don't like the world
We'll
anymore.
confuse everyone. I'll just do
Here we
it.
go.
Ready?
No,
Here we go.
No,
Oh, you're
I
gonna do
got it. 3, 2. This is TWIS. This Week in Science, episode number 1052, recorded on Wednesday, March 25th, 2026. Science Myths Defanged.
Great.
I got it. Yeah?
You got it.
Nailed it. Oh my gosh. Thank you, Blair. I was just going to let that one hang.
you
and
could
let
have.
it's just going to be one of those things that people are like, oh, it's a consistency error.
Yeah, there's 2 1053s. Weird.
Why? But it wouldn't have been because next week would have been 10.
Yours is the
Before.
funny.
I was just going to keep going. Bulldoze. Take a freight train. It's all good.
Yeah, for sure.
Let's see. DB. Justin is still a co-host, but we actually texted him tonight.
Yeah, he sent me back a photo.
Did he text back? Yeah. So we
Yeah.
were talking about this before the show started. To be
There he is.
Oh, he's working. Look
Yeah.
at that. He's all he's all labbed up.
He's lapped up.
That is clean room. Serious. Wow.
Yeah.
Yeah. So anyway, Justin, thank you, Paul. Justin, hasn't been with the show since towards the end of 2025 because he got a new job and the schedule conflicts with our recording time. And so it's a great job. I think it's been really good for him because it's funny. Yeah, exactly. All of it'll stay in the after show audio. Exactly, Paul Disney. It's not going anywhere. But yeah, we were missing him. And Blair's like, has he called you? Texted you? I'm like, no. Has he talked to you? She's like, no. I was like, let's text him. What's he doing? Yeah, he's doing his life, making sure that he's paying the bills and, taking care of his family and doing all this stuff, which is honestly the priority. As much as we love to have him here, if at some point he's able to shift his schedule, that's going to be great. If we need to shift our schedule because of life, we might have to do that. Like Wednesdays has been our time for a long time, but once upon a time we did Thursdays.
Yeah.
We
When I
also
started, we were on Thursdays. Yeah.
yeah, we have when I started doing the show, I think we were Tuesday mornings on
Yeah.
KDVS, so we've been all over the calendar. But what we need to do is just, find a time that works. And unfortunately, Justin's job is time shifted from ours. So he's a night worker now and not available as much in the evenings because that's when he's going to work. So, and Blair and I are day workers most of the time with people. Yeah, I remember Tuesday mornings. That was a long time ago, but that was before Blair's time even. Yeah, but where it all stands right now is that, like I said at the beginning of this year with Blair, like this is experiment year. We are going to do what we can to keep the, because we enjoy this, right, Blair? We like this.
This is why we're still doing it.
Exactly.
I would not be doing this.
I know.
When I have to wake up at 5 in the morning if it wasn't for fun.
Yeah, I mean, I have to get, that's me tomorrow morning. I have to get up for the conference. I have to be there at 7 because I have to get Camlink 4K to work with the freaking projector. And I would not do that today. Anyway, today was a fun day of tech difficulties. But yeah, we have late nights, early mornings, different schedules. And Blair now has a child and understands the pain that I've been in for a very long time. But yeah. But yes. Yeah, we're trying to keep it going, DB. Exactly. We are trying. And the fuel crisis, this, that, the other thing. I mean, we enjoy doing this and talking about science together with you and having this relationship with you, being able to talk about this kind of stuff. And so sometimes it's going to be just me. Sometimes it's going to be just Blair. Sometimes it's going to be the both of us. Maybe we'll get Justin back at some point for some stuff. Maybe we'll get some other guests in. Like sometimes we're going to have to cancel shows. Like it's just, we are keeping it going though, because we like it. We love it.
That's right.
Yeah. I don't want to lose this time with Blair. What would I do if I didn't get to hang out with you, Blair? Come on.
Aww.
Yeah.
Yeah, if I didn't have this outlet, I would have animal facts just spilling out of my ears.
While you're working in HR.
Yes, it still happens, I will say.
I have to say, if you are able to come up to Portland at any point this year, once a month, I'm doing a Brainy Birds class at this amazing arboretum, the White Arboretum. And it's just starting to be really fun.
Man, that's awesome.
Yeah, like it's hard. Like at first you're like, oh, I'm nervous and everything. And then I'm starting to like enjoy meeting people and talking about birds. And then we walk through the arboretum and go look for birds. And it's fun. You should come. It's awesome. Very geeky. But I mean, this is like another outlet, right?
Yes.
I'm getting paid for it, which is fantastic. But it's also a similar outlet. And maybe there's a zoo or a park outlet for some of your. Yeah. I don't know. There's arboretums.
Well, right now I'm telling my child about animal facts. How I'm getting to sleep these days is just telling him animal facts.
Oh, I love
Yeah, it's pretty fun. So he'll be like, "Tell me about the horned owl," and I'm like, "Okay."
You're
And
at
I
the
get to-
part where are you guys having conversations now?
Oh yeah. Oh, for
Of
sure.
course you are, but this is the best part, the toddler conversations where they're like,
They're really good. He asks me
They're
really
so
good
good.
questions too. And I'm like,
Mhm.
I'm teaching, we live right next to the bay, so I'm teaching him about estuaries. He can say
Mhm.
estuary, and we're like, I'm explaining what an estuary
How
is.
old
to
is he now?
him. 2.5.
And he says estuary. Of
He
course he
does.
does. Yes, okay. Yep. Of course he does. I love that.
I
But it's a
know.
little kid voice, so it's like estuary.
Mhm. It's really good.
I love it. It's so good. All right, everybody. I think Blair needs to go to bed so that she
Yeah.
can get up at 5 a.m. I need to
Mhm.
go away so that I can fix slides and make things for this video stream I'm doing tomorrow that was not working today. And maybe it'll be better tomorrow. Sometimes you just go back and it's fixed. We'll see. I don't know. It's fun. It's all fun.
Good luck.
Somehow I'm the hybrid virtual producer director
Mhm.
person, and so I
Makes
interface.
sense. Because you do this.
Yes, exactly. That's how it started, but now
I
I'm the
like YouTube
person who
video.
interfaces with the real world AV team. But then I'm like connecting everything with the tubes, and then
Mhm.
trying to make good experiences for people who are on the tubes
Mhm.
to see the things, and it's fun. I really like it. It'd be great to get paid for it.
Someday.
Someday. But
Someday.
honestly, my payment is building the community and making
There
everybody
you go.
happy.
That's right.
Yep.
You're making the world a better place.
Yep.
All
That's
right.
what I like to do. That's what I like to do. I hope everybody is going to have a better tomorrow or a better right now because of the fruit flies and mice and this TWIS experience.
That's right.
Okay. Are you not here next week or you are?
I'm here. I'm here next week.
Okay. I am not here next week.
Oh.
I have been invited to Washington, D.C. to be part of a National Academy of Sciences executive committee to put together basically like a policy brief of how we can pay for research and like make
Whoa.
the American research industry like be better.
Wow.
So that's where I'm going next week. And I'm going to be coming
Nice.
back Wednesday night, but I think I get back after 8 p.m. So, yeah. So I'm not getting paid for that either.
Sounds like another Oops All Animals next week.
Next week, Oops All Animals. Unless you want to have somebody join you as a guest also to talk Oops All Animals.
Oh, yeah.
Yep.
Huh. Okay. I'll think about it.
There's some good people out there. You could
Yeah.
bring them. Yeah. But anyway, I'm going to Washington, D.C. next week. I'm working with the National Academy of Sciences.
That's
I
great.
am person to help them frame the whole thing that we write.
That's cool.
I'm so excited. Like,
I can't wait to hear.
I cannot
more.
get sick this week at my conference.
Oh my god.
Is no, not allowed.
Yeah. Yeah. I can't wait to hear all about it and see the finished product. That's awesome.
Yeah.
Cool.
We'll see. Yeah.
Nice.
A ton of podcasts for 3 decades. Be our streamer. Exactly.
Yeah.
Yep. Ooh,
All right.
Natalia would be great for you to talk with. I love Natalia Regan. You should hang out with Natalia. She's like me, but a primatologist. Like, seriously.
Okay.
And she's a comedian who does burlesque and stuff. Like, she's fantastic. She thought a great idea. Yes, I could reach out and see. You guys.
Oh, I think I met her at Science Talk.
She's never been to Science Talk.
No, it wasn't Science Talk, but it was at the after party after our show that year at Science Talk, or I think I met her before.
I love her. She's great. I don't know if you have met her. We have had her on the show before, but I've like when you and Justin are gone, I've had her on like a couple of times. Just me and her. I have so much fun with her. Like,
cool.
She's fun. So Oh, you should get to know her too, because she does like Galapagos trips, and like
Mm.
she does like the cruises where you're the scientist who like tells people things over the cruise and stuff.
Ye-
I bet you would be amazing at that.
I would love to do that. I mean, I was
I
a tour
know.
guide for many years.
Yeah, like I could see you doing like I want you to be on one of those cruises.
That'd be super fun. I would love to do
You know? She's the one. Yes. That's such a great idea.
That's awesome.
I mean, I've hung out with her a million times. You should hang out with her.
Yeah.
Yeah.
Sounds great.
Mhm. Okay. Go to bed.
Alright. Yes.
I'm gonna go. I've got my energy up now to go
Oh, good.
do freaking slides. Come on.
Great.
It's always the last minute details. And then I have to get up at 5, 6, 6:30 if I shower tonight.
Oh man. Lucky.
Ah!
All right.
RPP says you should talk movie monsters and creatures. That would be fun. We do need to have like a TWIS Project Hail Mary watching. I think that would be
Oh
great.
yeah.
I
Yeah, I
really
haven't
want to
seen it
watch this.
yet.
Everybody
Yeah.
says it's great. And they're all mad about the centrifuge and then also alien languages. But I have to watch it. So anyway,
Great.
no
All
spoilers.
right. Good night, everybody.
Good night, everybody. Say goodnight, Blair.
Good night, Blair. Say good night, Kiki.
Good night, Kiki. Good night, everyone. Be well. Stay healthy. Get your sleep like I'm not going to tonight or for the next 3 days. And stay curious. Stay lucky. We'll see you next week.